Hello everyone. So in this high-level biology lesson, we will be talking about two of the most important organic compounds, carbohydrates and lipids. So to start off with, let's talk about the basics of carbohydrates. Monosaccharides are the building blocks of carbohydrates. They are the monomers that make up polysaccharides. And today we will be talking about four of these monosaccharides. The first one is glucose. Glucose is the form of sugar that fuels respiration, and it's the base unit for many of these polysaccharides. So glucose is a hexose sugar, meaning that it has 6 carbons in its structure. You can see 5 of them in the ring and 1 on the outer part of the ring. So moving on, the second monosaccharide is galactose. Galactose is also a hexose sugar, but it's less sweet and it's most commonly found in milk and cereals. So galactose and glucose have the same formula, but there's still structural differences between them, as you can see in this photo. So the third monosaccharide is fructose. And fructose is a pentose sugar, unlike glucose and galactose, meaning that it has 5 carbons. And fructose is commonly found in fruits and honey, so it's the sweetest naturally occurring carbohydrate. And the next monosaccharide and final one that we'll be talking about is ribose. And ribose is also a pentose sugar, and it has a pentagonal ring. So here in this structure, not all of the carbons are labeled, but all of the numbers mean that there's a carbon there. So there are 5 carbons in ribose, meaning it's a pentose sugar, and ribose forms the backbone of RNA. We can similarly think of deoxyribose, which forms the backbone of DNA. So monosaccharides are the monomers of polysaccharides. So these sugars like glucose and galactose and fructose, they go through a condensation reaction to bond and create a disaccharide or a polysaccharide. So in this reaction, we see that 2 glucose monomers have joined through a condensation reaction where water is removed and form the glycosidic bond, creating maltose, a disaccharide. And water is a product of this reaction. So now let's talk about the types of disaccharides. So maltose is the first one that we will be talking about. Maltose is the product of glucose and glucose. And maltose can be commonly found in wheat, corn, or barley. As we can see, these 2 glucose monomers are joined together by a glycosidic bond. And the next disaccharide is lactose, which is a product of galactose and glucose. So lactose can be found in milk. The third disaccharide is sucrose, which is also known as table sugar. Well, sucrose is made up of glucose and fructose. And since we know that fructose is the sweetest naturally occurring carbohydrate, we can infer that table sugar can only be made out of fructose and glucose, of course. So moving on to polysaccharides, which are polymers consisting of more than 2 molecules. They're very long and might be branched, and we will be talking about 3 types of polysaccharides today. But before we move on to the types of polysaccharides, first let's talk about the 2 types of glucoses that we might encounter, which are alpha glucose and beta glucose. So the only difference between these 2 is where the OH is located. So in alpha glucose, OH points downwards, and in beta glucose, OH points upwards. Okay, so now we can move on to the types of polysaccharides. So the first polysaccharide is cellulose, which is made by linking together beta-glucose molecules. So carbon 1 and carbon 4 are linked to each other, and glycosidic bonds are formed. So as we can see in this picture, the glucose subunits in the chain are oriented alternately, like this. And this causes the cellulose molecule to be straight. So it cannot bend because there are subunits in each side of the chain. So it's a straight molecule and it's unbranched. So this gives cellulose high tensile strength. And the tensile strength prevents the cells from bursting under very high pressure. And this is why cellulose is a very important part of cell walls in plants. Moving on to the next polysaccharide, which is starch. So starch is made by linking together alpha glucose molecules, and carbon atom 1 and to carbon atom 4 are linked. So first let me explain what we mean by carbon atom 1 and carbon atom 4. So in one of the previous slides, I showed you how carbon isn't always written on the structure, but it's shown by numbers. So this is how we number our carbons in our glucose molecules. So as you can see here, carbon atom 1 and carbon atom 4 are linked together. So that is what we mean by this sentence. So as we can see in this picture, unlike cellulose, all of the subunits are on the same side of the chain in starch. So this means that the starch molecule is curved, it can bend. And there are also two forms of starch, amylose and amylopectin. So amylopectin has a branched structure. It can be much larger than amylose. It can consist of 2,000 to 200,000 units. But amylose is unbranched, and it forms a helix instead of a globular shape. So amylose is much smaller than amylopectin. So let's talk about the function of starch. It's only made by plant cells, and it is the storage of energy in plants. So glucose is held as starch in seeds and storage organs such as potato cells. Now let's move on to our final polysaccharide, glycogen. So glycogen is not a simple chain, it branches many times, and condensation reaction links carbon atoms 1 to 4 on the alpha glucose, but when it branches, it links carbon atoms 1 to 6. So this causes it to be very large in size, so it can be up to 30,000 units, which is a lot. So the main function of glycogen is it is the storage of glucose in animal cells. So it's stored in the liver and some muscles in humans. And glycogen is a useful energy resource. It's a fast energy resource for animals. And this is the structure of it, as I said. And so we can see, let's number the carbon molecules here quickly. So carbon atoms 1 to 4 are linked here. But here, we can see carbon atom 6 is being linked. So this glycogen forms branches these are the branches, aside from the main chain of glucose. So let's move on to the next organic compound, which are lipids. So first, let's talk about the three types of lipids. The first one is triglycerides, which are made up of 3 fatty acids plus glycerol. So triglycerides can be found as either fats or oils. So by fats, for example, butter, or for oils, it can be sunflower oil or vegetable oil, things like that. And the second type of lipids is phospholipids, which are found in the cell membrane. And the final types of lipid is steroids, which can either be cholesterol or sex hormones such as testosterone or estrogen. So these are the three types of lipids. Now, let's go back to the basics. Let's talk about the general structure for a fatty acid. So we said that triglycerides are made up of 3 fatty acids, and this is the structure of those 3 fatty acids. So we have a chain of carbon and hydrogen atoms, so it says N because we don't know how many there are. And we also have a hydroxylic group. And there are 3 types of fatty acids. So the first one is saturated, which has no bond, and this is how it's shown. The second one is monounsaturated, which is shown by this little line here. And the final one is polyunsaturated, which has more than one double bond. So this signifies a double bond. So this has 2 double bonds. So it's polyunsaturated. Also, firstly, let's give an example to a saturated fatty acid. So this can be butter, for example, because saturated fatty acids are solid in room temperature, and unsaturated fatty acids are liquid in room temperature. So this can be olive oil, for example. Okay, so yeah, so there's saturated, monounsaturated, and polyunsaturated, and this has double bonds. This is the important detail. So here's an example. So we have 3 types of fatty acids here, and we need to figure out which one's saturated, which one's monounsaturated, and which one's polyunsaturated. So let's look at the first one. Here we can see that it has 1 double bond. So what did 1 double bond mean? It meant monounsaturated, because why? Unsaturated fatty acids have a double bond, but since it has only 1, we can infer that it's monounsaturated. So moving on to the second one. So here there are no double bonds. So we can say that it's an unsaturated fatty acid. And the final one, we have 1 over here and another over here. So we have 3 double bonds, meaning that it's polyunsaturated. Also, there are 2 types of fatty acid isomers. So these are types of unsaturated fatty acids. So they're either cis isomers or trans isomers. So first, let's take a look at the structure. In cis isomers, we see that 2 of these hydrogens, they're at the same side. So this means that they can be bent because there's nothing here that's preventing them to bend. So we can say that the double bond causes a bend in the fatty acid chain. So this is the double bond, by the way. However, let's look at the structure of trans isomers. So we can see 1 hydrogen atom here and 1 here. So since they're on alternate sides, this means that this molecule cannot bend. So the double bond does not cause a bend in the fatty acid chain. So the most important difference between these 2 is this. And we can say that cis isomers are found very commonly in nature, but trans isomers are very rare. So this means that they're artificially produced. For example, margarine is a trans fatty acid. So this means that cis isomers are healthy, while trans isomers are bad for our health since they're not found in nature. And so we said before that unsaturated fatty acids were liquid in room temperature. However, since these do not bend and they're artificially produced, they are solid at room temperature. Again, margarine is a very important example of this. Okay, now let's move on to another question. Which one is trans? Which one is cis? So let's look at the first one. It does not bend. It has a double bond, but it does not bend. So this means that it's a trans isomer because since it's very densely packed and the hydrogen atoms are in alternating sides, it cannot bend. But here we see the double bond and it does bend. So this means that it is a cis isomer. Another question. So yeah, again, we see the double bonds. This is also polyunsaturated. We see the double bonds and we see the multiple bends here. So again, this is a cis isomer. Yes, cis. So let's look at one of the most important types of lipids, triglycerides. So as we said, it's made up of glycerol plus 3 fatty acids. So here are our 3 fatty acids and here's our glycerol. So what was the reaction that combined 2 monomers and made them into a dimer or a polymer? It's condensation reaction. And during condensation reaction, we need to take water from the existing monomers, and water is produced at the end with a triglyceride. So here, 2H and 1O is taken, and these two are bonded, and this bond is called an ester bond. And in the end, a triglyceride and 3 waters is produced. Now let's talk a little bit about the functions of lipids. So we talked about the 3 types of lipids. One of them was phospholipids. So phospholipids play an important structural role because they're the main component of cell membranes. And steroids are involved in hormonal signaling since they are sex hormones like estrogen or testosterone. And other functions of lipids are, first of all, insulation. So fats in animals can serve as heat insulators. Think of bears or polar bears. So the reason that they can survive in such cold weather is because they have a lot of fat in their body, which insulates them and keeps the heat. So, and also protection. So triglycerides form a tissue layer around our key internal organs and provide protection against physical injury. So our belly is there to protect us, actually. And finally, storage of energy. So triglycerides are a very important long-term energy source. Think of bears who hibernate, for example. They can sleep all winter because they have this incredible amount of energy stored because of all the fat that they have eaten. However, now let's compare carbohydrates and lipids in terms of energy storage. So lipids are used for long-term energy storage, while carbohydrates are used for short-term energy storage. This is because lipids contain more energy while carbohydrates have less, but the lipids in our body are stored in the adipose tissue, which is located beneath our skin and around some organs, such as our kidney. Even though lipids are a very important energy source for us, Fats in adipose tissue cannot be mobilized as rapidly as carbohydrates. So glycogen, which as we said was a polysaccharide that was the form of storage of glucose in animals. Glycogen can be broken down into glucose rapidly, and it can be easily transported, while fats take a longer time to utilize. So here's an analogy so you can understand. So we are paid in cash, which is energy. And if we need to use it quickly, which is we do respiration, we use glucose immediately. But if we want to just keep it where we can access it quickly, we put it in our wallet, which is easy to get to, it's easily accessible, and we can store a little amount of energy, money or energy, but we can use it quickly. So it's a fast way of reaching that energy. But lipids are like storing the money in the bank, because taking it out takes a long time, but we can store a lot of money, a lot of energy. So the final thing we will be talking about today is body mass index. So body mass index is a screening tool to identify possible weight problems. So it is calculated by dividing our height in meters squared by our mass in kilograms. And the result falls somewhere under this category, which determines our status, such as overweight, normal overweight, or obese, etc. So let's solve an example for BMI. So it says a man has a mass of 75 kilograms and a height of 1.45 meters. So how do we calculate his body mass index? We divide his mass in kilograms by his height in meters. So let's quickly do. So his body mass index is 35.6. So let's check what that means. So here, let's look. So it says 30 and above falls under the obese category. So this man is obese according to the body mass index. And what can we say about the relationship between height and BMI? So as height increases, BMI decreases, because as the denominator increases, the overall results will decrease. Yes. You can solve these questions in your own time, and here are the answers. So yes, thank you for listening to this lesson. I will be posting the resources under the video if you want to take a look at them, and I will be posting some extra notes as well. So thank you for tagging along.
Bye.